It's the first time I've had a round of applause before I've spoken. Thanks for inviting me. As said, I'm Phil with 2 L's. I work for the Washington Department of Fish and Wildlife, and I am the lead for our forage fish research and management team. I've been doing forage fish research for about 9 years now. I sort of accidentally fell into forage fish research because I was actually doing research on sturgeon spawning. So sturgeon are much, much larger than the fish we're going to be talking about today. But I was looking for sturgeon eggs and absolutely failed at finding them during my master's thesis, but I actually found a whole bunch of smelt and other sorts of forage fish eggs by happenstance, and moved to Washington from the East Coast to do sturgeon research, but sort of got pushed into doing some additional forage fish research, and ultimately that became my job. But today, I could talk all day about forage fish, but I have to keep it down to sort of a limited area of topics. So just for those of you who may not be aware, I'm going to go over what forage fish are and why they're important. And I'm going to introduce you to some of the species that we manage here in Washington State, and also how we study forage fish. And we do a lot of different research projects working with different agencies and NGOs and community groups. And I'm just going to highlight sort of one of these different studies from different parts of the ocean on different species. Hopefully you sort of see the width or the breadth of different research that we do and techniques that we use through this. Like I said, I can talk all day about forage fish. So if there's something that really interests you, I'd rather go off on a tangent and talk about that. So feel free to ask questions as we go or comment. If it's something I plan on getting to later, I'll let you know. But if it's something that we weren't going to talk about and you're interested in it, I'd rather talk about what you're interested in than just letting myself ramble all day. So to begin with, what are forage fish and why are they important? Forage fish are an ecological group as opposed to a species or genetically related group. So sometimes we'll even call them forage species because we might include things like krill or squid in that ecological group. But for our management purposes, we're talking about the generally small, shiny fish that are schooling in the ocean, generally very abundant, highly fecund, meaning they produce a lot of offspring or eggs. And they fill this middle tier of the food web, and they're a vital conduit of energy from these lower level producers and consumers to these higher level consumers. And this is what we call our wasp waist or hourglass food web model. And the shape of the food web model indicates the diversity of species at each level. So there's sort of a bottleneck here at the forage fish level. And so if we lose one of these species, the entire ecosystem becomes less robust. But I like to always point out that on this upper end, where all the things that eat forage fish are, we have whales, we have salmon, we have rockfish, we have seabirds, we even have people. So I always like to put a plug in there. If you like whales or salmon or rockfish or seabirds or people, then you should probably like forage fish because all those things at some level are relying on forage fish for their food. Forage fish are also an indicator of ecosystem health. Because of this vital role they play in that sort of middle tier in the food web, if things aren't going well with forage fish, it generally means things aren't going well for other species. So we've seen instances in Washington State where rhinoceros auklets have died off, and there's been suspicion that has to do with prey availability, their prey often being these sand lance, which are forage fish. Harbor seals also feed on forage fish. And another thing that I think is sometimes overlooked, and I hope to talk about sort of at the end to wrap everything up, is that forage fish are not only important as forage, the term forage fish, we use that because they're forage for other species, but sometimes they actually act as a prey buffer. So these are forage fish down here. Typically we don't consider these forage fish because these are salmon smolt, they're juvenile salmon. But if you're a hungry harbor seal, these guys and these guys look pretty similar, so if there's not enough forage fish around, these might be the next thing on the menu. And finally, forage fish are both culturally, recreationally, and commercially important. All of these images are from a pretty iconic fishery here in Washington State. This is the Yulicon smelt fishery, and it's culturally important. This is the rendering of Yulicon for their grease. They're sort of a unique species, and their fat is actually a solid at room temperature, so their grease would be rendered and traded by First Nations and tribes, led rise to what was called grease trails, trade routes across the Cascades. I've even heard rumor that the name of the state of Oregon might be a derivative of the word oolican, which was what the natives called these fish. I'm not sure if that's true, but it's an interesting story. So these have been really important, and we're going to talk more about oolican in the future. But globally, forage fish account for over a third of marine harvest by weight. So they're pretty critically important. Interesting note though, not all forage fish, actually most forage fish aren't used for human consumption. Generally they're used for things like feed and fertilizer or even bait. And sometimes they even like to put out like, well, might be a vegetarian, so I don't need to worry about forage fish. Well, sometimes that fertilizer is actually from forage fish, so even vegetarians might actually be benefiting from forage fish fisheries. So, introduce you to the forage fish that we have in Washington State. This isn't all of them, but these are some of the common ones. And sort of simplicity's sake, I've broken them down into 3 groups based on where they spawn. The first group I'll show you are pelagic spawners, meaning that they spawn out in the open water. So their eggs are sort of adrift after they are spawned. Anadromous spawners, which are fish that will grow in the open water and then return to rivers to spawn, similar to salmon. And then nearshore spawners, which are pretty critical in Puget Sound because of all the shoreline we have and the human development we have. So 3 examples of our pelagic spawners are the northern anchovy, Pacific sardine, and Pacific mackerel. Again, these are open water spawners. Generally in the Salish Sea, we don't see these bottom ones too often. It's generally the northern anchovy that we see more commonly. Our anadromous spawners are that oolican smelt that I mentioned previously, and then also the longfin smelt. So these, the oolican typically are coming back this time of year actually. There's actually just a small recreational fishing opportunity yesterday down on one of the tributaries to the Columbia. And they'll spawn and typically die after spawning, similar to many of the salmon species. And then where we spend a lot of our time and resources studying are what we call our nearshore spawners. And the reason we'll learn more about that we focus on these so much is because of the impacts that humans are having on the nearshore in Puget Sound. These guys are pretty susceptible to those, and so we try to figure out where they are and when they're spawning. But we have in the lower intertidal and subtidal spawning areas our Pacific herring, intertidal spawning sand lance, and then also our intertidal spawning surf smelt. So quite a range of different life histories across forage fish means that they face different challenges throughout their life histories, and that we also have to use different techniques to study them. So some of the challenges that are facing forage fish are sort of the usual suspects, if you will. If you believe it or not, climate change is actually a threat to forage fish. The reason for this is that some species of forage fish seem to thrive when we are experiencing warmer ocean conditions, and others seem to thrive when we're experiencing colder water conditions. So you can imagine that if we're continually having longer, warmer ocean conditions, that those species that thrive during warmer water conditions might be more abundant, might be more widespread, while the fish that prefer the colder conditions might be less widespread or less abundant, which overall seems like it may not matter so much, except for that makes for a less robust ecosystem. And if we ultimately lose those colder water fish and only have the warm water fish, if we do happen to have a cold spell and those warm water fish drop off, there's nothing left to replace them. And so we like to see sort of that nice mix of several species so that even though one species might be down, the other will be up. Overfishing has been a problem for forage fish, particularly in the past, because forage fish have these natural fluctuations in abundance. We call them boom and bust cycles, where sometimes the abundance is way up and then other times it's way down. So it makes them really hard to track their abundance, but also because of their schooling behavior, forage fish can be pretty easily targeted by purse seines and other fishing equipment. You can even use spotter planes to see these schools of fish. And as a result, as fishery managers are monitoring how many fish are being caught, they don't see a decline in the catch rates until after the fishery has already been depleted. And so it means that one of our primary fishing tools, catch per unit effort, isn't very effective for monitoring these fisheries sometimes. Also, because so many things like to eat forage fish, they're sometimes associated with other species that are being targeted, and they get caught incidentally as bycatch. And then the last two, poor water quality and habitat degradation, are ones that we're particularly concerned about here in the Salish Sea and the Puget Sound, because again, we have so much shoreline and so much human development over a relatively small water area. So there have been some studies that have looked at the effects of water quality on forage fish, particularly on their embryos. I've heard some of the researchers say that every time we have a big storm event, all the runoff is almost like a mini oil spill, and those eggs get the brunt of it because they're in that nearshore environment. Yes?
Just got one quick question about the overfishing / bycatch of them. So obviously, certain fisheries, you have regulations and legislations for, the size of the fish, or if it can be like the male or female, etc. But with forage fish, especially being caught in, purse seines and all that good stuff, it's much more easy to grab every single fish there. It's really hard to, to piece out the specific ones. Just out of curiosity, what are sort of like the regulations and legislations that protect the forage fish and, their reproduction and all that good stuff?
Great question. So in Washington, we only have, inside Washington waters, we have some pretty minor fisheries. We have a herring fishery, and we're very conservative about managing that fishery because of the recognition of how important it is to the ecosystem. And so one thing that we do is that we limit the catch as to a percentage of our biomass estimate for the spawning stock biomass. And we set that pretty low. So for example, in Alaska, I believe their threshold or their guideline is about 20% of the total population. In Puget Sound, we say 10% of our spawning stock biomass estimate, which the spawning stock biomass is only a fraction of the total population, and then we further limit it to areas where the fishery is occurring, so we reduce it even further than that. And we close the areas where the fish are actually spawning to avoid those spawning grounds during the spawning season. So we try to be really conservative about how those fisheries are managed to ensure that we're not having impacts up the ecosystem. So inside Puget Sound, again, water quality and habitat degradation. Those nearshore spawners, we're really concerned about that habitat degradation as well, because lots of people like to be on the beach, and as a result, they build their houses near the beaches, and that has an impact on how the beaches function. And we'll talk a little bit more about this, but the surf smelt and the sandlands in particular have preferences with regards to shading on the beach and sediment composition on the beach. And if we're essentially taking away that habitat, those fish no longer have a place to spawn. Oops. So how do we study forage fish? Now there's just a question about the fisheries, and that is, historically the primary way that we study and manage a lot of fisheries is by what we call fisheries dependent monitoring. So from fisheries dependent monitoring, we can get something that we call catch per unit effort, essentially how many tons of fish do you catch per unit effort, which could be a number of net sets or soaking time for a river or for a net, or a number of hooks on a long line, all sorts of different metrics that we can use for that. We can also get stock composition data by biologically sampling, or biometric data based on the length or the age of the fish. So, and this is really still very critical, but as I just mentioned, we keep our fisheries pretty small in Puget Sound, in the Salish Sea, southern Salish Sea, and also a lot of our fisheries are closed, so we don't get this data as readily as we could, or those fisheries are so limited we don't get representative data anymore, which means that we have to use what we call fisheries independent monitoring. And that's mostly what I'm going to talk about today, is a few different forms of fisheries independent monitoring that we use in order to get information to sort of manage and maintain these stocks and monitor where they're going. So sort of starting at the big scale and at that pelagic scale, the first technique I'm going to talk about is our coastal pelagic species surveys. And they use what we call an acoustic trawl method. It's a two-part method in which this is actually the Reuben Lasker. It's one of the most advanced research vessels in NOAA's fleet. And this map on the side here, all these lines represent transects that they do all the way from the northern tip of Vancouver Island all the way down to the border with Mexico. And they do these acoustic trawl surveys in which the boat is running fancy sonar and it identifies schools of fish, and then it comes back with a trawl net and targets those schools of fish to capture the biological samples to figure out the species composition, size, age, etc. And it also is estimating the biomass based on the size of these schools. So they get a density per area, essentially, of the school size, and so they can back calculate from the composition from the trawl data to the number of fish they expect, or the tons of fish they expect would have been there for that acoustic signal. And there's all sorts of cool data that gets out of this, and we collaborate with NOAA to do this. This is actually me right here. I got to go out for 2 weeks on that vessel, the Reuben Lasker. It's actually a really great experience. The only downside is that the way they run their surveys is that forage fish typically will go deep during the day and then come to the surface at night. So they run their acoustic surveys 12 hours during the day, and then they do their biological sampling 12 hours at night. So I had the 8 p.m. to 8 a.m. shift. And there's always a crew running 24 hours a day. There's 2 lead scientists, and they're rotating 12-hour shifts. So it's around-the-clock sampling using different techniques. So yeah, it's sort of interesting to be on the night shift for 2 weeks and then come back to land and try to get things sorted out. But we get the stock composition, or a lot of information from that. This is a tray of, I believe those are Pacific mackerel being prepped for sampling. We'll take information about otoliths to get, or we'll take samples from otoliths to get their ages, all sorts of biological data that are gathered during these surveys. And more recently, we've also begun working with NOAA and some of the industry partners on a collaborative study to expand this sampling. So one of the limitations of such a large vessel like the Reuben Lasker is that it can't get very close to shore. So working with this vessel, the Lisa Marie, NOAA outfitted the vessel with the similar acoustic system that they have on the Reuben Lasker, but the Lisa Marie is much smaller, so it can get into much shallower water. And so these pink dots indicate where the Lisa Marie was augmenting those surveys near shore to get into those shallower waters to see if there was any more fish that were being missed by that survey. And this just is, the Lisa Marie is a purse seine vessel, and so they also sampled fish using the same gear that the commercial fishermen use. And one of the advantages of the purse seine vessel is that, unlike the trawl, where essentially all the fish that come on board are assumed to be dead because they sort of get squished into that net, not always the case, but usually, the purse seine can close up around a school of fish, take a few dips out with this long-handled dip net, take the samples we need, and then open the net back up and let the rest of the fish go. So there's virtually no harm to the fish that aren't brought on board. And all this information is compiled to go into these stock status reports that NOAA provides. And this is just an example of, for the 3 species I spoke of before. The orange line is the Pacific sardine. You may have heard of Cannery Row down in California. There, these sort of iconic fisheries that collapsed. There was a bit of a rebound here in that fishery. We also have the northern anchovy in green, and then the Pacific mackerel in black. The northern anchovy is less complete because sometimes there's just not enough resources to do the surveys, and sometimes the abundance isn't low enough to get a good estimate. So sometimes challenges with these. And going back to that question about how we manage these fisheries, this is one way that these fisheries are managed. This is a larger scale fishery, but there's now a biological reference point where essentially the fisheries managers have said, if our estimates of sardine biomass go below this point, we're just closing the fishery. And so that's what they did back in 2015. They had very conservative fisheries, and they continued to go, the stocks had increased during the fishery, but then it came down again. Again, those dramatic fluctuations. We don't think that this was because of the fishery, probably more related to ocean conditions, but regardless, they shut down the fishery. So I mentioned that those large boats have limitations. They can't get very close to shore. Well, obviously, they're also not going to come into the rivers. So how do we sample for the anadromous species? So we can use some similar techniques for anadromous species, but for the Eulicon, we've developed a method, actually I should say the Canadians at Department of Fisheries and Oceans developed a method to sample ichthyoplankton or larval fish using a plankton net to estimate the abundance of the eulachon returning to the rivers to spawn. So these are eulachon smelt, and these are baby eulachon smelt. They're about the size of an eyelash. They're only about 7 millimeters in size, and they have eggs that are adhesive, so when they're spawned in the river, their eggs sit on the bottom for about 4 weeks attached to sand and gravel. Then they hatch out, and they're pretty quickly washed out of the river. And so as they're being washed through the river, we have several transect sites where we set up these plankton nets. We'll deploy them 1 to 2 days a week. And in this plankton net, there's a flow meter. So we know the amount of water passing through that net. We know the area that the net is sampling. So we can estimate the volume of water that we've collected samples from. And then we actually take those back to our lab and count all of the eggs and larvae that we find in there. And from that, we calculate the total flow through that river system for that period of time to back calculate the number of larvae, back to the number of eggs, back to the number of females it would have taken to produce that number of eggs. And then we multiply that by 2 because there's males and females. It takes 2 to tango. And then we come up with our biomass estimate. It seems pretty complicated. Yeah, question?
I just have a question over the previous slide. What is anadromous?
Oh, sorry. So anadromous is like salmon. So they spawn in rivers, but then they move out to the ocean to grow up. They become adults in the ocean, then they return to the rivers to spawn. So it's just a term for their life history. And so we've been doing these types of plankton surveys or ichthyoplankton surveys since 1996. And the reason that we started doing them in 1996 is that in 1994, we saw a pretty dramatic decline in the landings from the commercial fishery in the Columbia River. And so if I had showed you a graph, this gray area here is the landings, essentially an estimate of catch per unit effort, if you will. It's usually a pretty good index of abundance. And before 94, it was much higher. It was actually would have been like up here in the early, or before 94. So because of this decline in landings, the fishery had to be curtailed, and we were no longer getting good information from the fishery. So we began using this ichthyoplankton net, and this is our density estimates for larvae coming out of the Columbia River. And you can see it tracks really well with our catch per unit effort estimates during the same time frame. So this gives us an indication that those larval estimates or densities correlate really strongly with our fisheries data and is probably a pretty good estimator of the relative abundance of fish in the river when we're using those surveys. And then in 2010, we don't have any more commercial data because the eulachon smelt was actually listed as threatened under the Endangered Species Act because it had been declining for such a long period of time. So really now this is our primary tool, these ichthyoplankton surveys, is our primary tool to estimate the abundance of eulachon in the Columbia River. Oh, and I can't go on without also pointing out that one of my favorite things about eulachon is that one of their common names is candlefish. And the reason they're called candlefish, obviously, is because they are so fatty you can actually dry them out and light them on fire. Question?
Are there any runs between Columbia and the Fraser River?
There are. The Chehalis River gets a run. I believe the Quinault and the Queets and the Elwha. But by and large, the Columbia is an order of magnitude larger than even the Fraser run. So it's it's the largest run in our region.
How are they doing in British Columbia?
British Columbia have seen similar declines, and that was one of the reasons that the NOAA opted to list them, was because it wasn't just a single system decline, there's a whole region that we've seen declines in. And so again, most of the fisheries targeting eulachon have been in the river, but since we've seen regional declines, that's another indicator that we don't think this is a fisheries-driven decline. It's much more likely that it's something happening in the ocean. All right, so we've spoke about how we do some surveys out in the open water. We've spoken a little bit about how we do some surveys inside of rivers. Now I'm going to talk about how we do some surveys for those nearshore species in Puget Sound. So this is actually what we call a whitewater event over in Hood Canal. This was just last year in April. And this is a mass spawning event of herring. So herring deposit their eggs on submerged vegetation or 3D structure, typically things like eelgrass or algae. And we use that that life history strategy and approach to spawning in order to help us estimate their abundance. So currently, our primary approach to estimating herring abundance is what we call rake surveys. Essentially, this is an estimate of the density of eggs in a given area multiplied by the area to then calculate to the number of adults it would have taken to spawn that number of eggs. And we use small vessels because we need to get close to shore. And we actually use this rake. It's called, we call it a rake. It's essentially a vegetation grapnel that we just drop to the bottom and drag for a few meters to collect clumps of vegetation. And this is what herring spawn looks like when we pull it up. So they're deposited on different types of algae. And then we have a key that we use based on the densities where we'll say, either the eggs weren't present or they were light or very light all the way up to very heavy. And there's actually a number that's associated with that, with a lineal distance of density across the shoreline that we'll use as an expansion factor to estimate how many tons of herring would have spawned in that area. And we've been doing herring surveys since the 1970s. We started tracking herring. And over time, this map has added more stocks to it, but we've identified at least 21 different spawning areas inside of the southern Salish Sea that we monitor every year. Spawning is occurring right now. It typically begins the first week of January and extends through mid-April, but there are a couple of special stocks that may spawn all the way through June. So this is really our busy time of year. Even though we call them stocks, they're groups that are consistently coming back to the same areas to spawn during the same time of year. Genetically, we've only been able to identify 3 different groups of herring in Puget Sound. On the south end, our Squawks and Pass stock is a genetically distinct stock. On the north end, our Cherry Point stock is genetically distinct. And then all the other stocks in between are lumped together as this other stocks complex, is what I like to call it. So this figure here is our biomass of those spawning stocks going all the way back to the 1960s. The dotted lines just represent periods when our sampling was less consistent, so there might be some rounding area. And this is the 4-year rolling average, which is one of our preferred ways to show the abundance because there is so much variability from year to year. So we like to average over 4 years to sort of see what's going on. And what really stands out here is this Cherry Point stock. It used to be the most abundant stock in Puget Sound. Cherry Point by itself was more abundant than all the other stocks combined in Puget Sound. And it's declined by over 98% since the 1970s. And we really don't have a good idea of why that is. But there has been some research looking at, or at least some researchers that have discussed, where the different trends are being seen. And one thing that I think sort of stands out is that when we look at the Salish Sea as a whole, populations of herring has actually been up in recent years because the northern Salish Sea in Canada, those stocks have been doing very well. But they've also seen declines in their stocks sort of in the southeastern part of the Strait of Georgia. And it seems sort of telling to me that the closer you get to the population centers, it seems like the less well these herring stocks are doing. So something to keep in mind. Again, we're not quite sure why, but that seems to be a, sort of something that stands out when you look at the larger regional scale. We also, for those other forage fish that are spawning in the near shore, these are actually fish that will come up on the beach and deposit their eggs at a high tide, and the eggs are left there as the tide goes out. So these are surf smelt eggs. Surf smelt are obligate intertidal spawners. They're about 1 millimeter in diameter. This is a pretty heavy spawn event. But to do these surveys, well, actually, I should say, we don't do these surveys to estimate abundance. The purpose of these surveys is actually to identify where and when spawning is occurring. And the reason we want to know that is because we want to protect that habitat from things like this. The type of gravel and the shading that's on the beach plays a key role in whether or not the eggs deposited on the beach actually survive. So when the tide goes out, those eggs are left high and dry. And if you have the right mix of sand and gravel and some shade, it will stay cooler and more moist on that beach, and we have lower mortality on those eggs. So the composition of the beach is pretty critical. But things like this is what we call a direct habitat loss. Before this was built, forage fish would have spawned up here. Obviously they can't anymore, so that's just direct habitat loss. It's gone. But we also see different types of habitat loss that are more indirect in the sense that they affect beach processes. So a structure like this is preventing sediment from being eroded from the uplands and down onto the beach. And in Puget Sound, most of our beaches are created from the erosion of the uplands. And so those beaches are going to get shallower, and the sand and gravel is going to get washed away, so they're going to get harder, and it just becomes a less suitable habitat. We also see clear cutting of trees along beaches because people want to see the water, they don't want to see the trees, and so that reduces the shade, and that also has a negative impact on the habitat as well. So to do these surveys, we actually just go out and collect bags of sand. We don't even need to get our feet wet really to do these surveys. But then we reduce that bag of sand down to egg-sized material to reduce the volume of it. So we're starting with about 2 liters of sand to begin with. And then we do a process called winnowing to get the lighter material, because the eggs are typically attached to sand and gravel, but they'll be a little bit lighter than the sand and gravel they're attached to. So if we can agitate them to come to the surface, then we can extract them more easily. And then we have to go into the lab and use a microscope to actually find and identify those eggs. Now in the past 5 years, we've been using a new method that we've developed, which has really sort of revolutionized our approach to sampling. And we call it the vortex method. So if you're not familiar with it, it's using this gold panning device and creates a vortex, sort of a whirlpool. And we call it the vortex method because it's got a V and an X in it, which means it's awesome. So we did some extensive testing where we actually took known numbers of eggs and put them into these samples. And then we had novice users try them out and see which one worked better. And what we found is with the old method, we got about 60% of those eggs back, which is pretty good. But with the new method, we're getting about 90% of the eggs back. And it was much more consistent across users. So people that know how to do this can do this very well, but everybody could learn how to do this. So we've shifted over to using this method. And really, what's been even more impressive about this is that previously, we had to sort through this much sand and gravel to find those 1 millimeter diameter eggs. Now we sort through about this much sand and gravel to find those 1 millimeter eggs. And there's actually more eggs in this sample than there is in this sample. So it means that it's much easier to find those eggs, and it takes much less time. The caveat to that being that sometimes this method is so much more efficient that we have so many more eggs that it'll actually take us longer to process a sample because we have to count to a higher number. But otherwise, we think it's a big advantage because it means that we're not missing eggs as much as we used to. And what we do with this information is what's really important. This information is all updated online, and it's provided to habitat planners and habitat biologists, the folks that have to make decisions about how our shorelines are used. And so they can look at this online map and say, well, geez, there's some herring spawning here, and there's some surf smelt spawning here, and there's some sand lance spawning here. Maybe we should change our approach to how we're managing this habitat since it's so critical to so many species. And that's what we like to see. And we've been able to partner with groups like the Northwest Straits Commission. DNR has been funding a lot of this research with the Washington Conservation Corps. And we've been working with a lot of marine resource councils to help us collect this data because we are so confident in those methods now. So that's about all I want to talk about with methods, but I think I just have a couple of minutes here that I want to share with you something I think is really cool about sort of tying everything together. So in about 2015, we started seeing big schools of fish, particularly in South Puget Sound. These are actually anchovy that were seen by eyes over Puget Sound. And we actually started seeing them during some of our other studies using beach seining. And this is what about 250,000 juvenile anchovy look like in a 100-foot beach seine. And we've been seeing this pretty consistently when we're beach seining in October since 2015. And a few years ago in South Puget Sound, we also started seeing large aggregations of marine mammals. So several hundred, 200 to 300, sometimes up to 500 sea lions have been showing up in South Puget Sound, where previously there might have been 50 to 100, and they're gorging themselves on these anchovy. There's even common dolphins coming in there, and they'll be in there feeding on this big schools of anchovy for several months. We've estimated that they're probably consuming a couple 100 tons of anchovy during the time frame that they're down there. So with all these marine mammals down there eating these forage fish, eventually they catch the attention of another marine mammal. This isn't your southern resident killer whale. These are the transient killer whales. And it was really, really interesting to us last year that in mid-March, after we had observed all these sea lions, one day we were going through the area and all the sea lions were gone. And lo and behold, transients had showed up and essentially chased them off. But what I think is even cooler about this is that we've got really limited data about anchovy in Puget Sound. We know that they're very flashy. So essentially there's periods of time where we can't find them and then they'll erupt and we've got this big boom. And so, like I said, we saw this beginning in 2015 and they've been pretty high for a while. And we started looking around for some other data sets that might support this. And sure enough, the Strait of Georgia Age 0 herring surveys had information that said, yeah, back in the mid-2000s, they saw a lot of anchovy. And then the same time frame that we're looking at, they also saw more anchovy. And we started talking with some of our colleagues that are doing salmon studies. And during the same time frame, there was a really cool tagging study in which juvenile steelhead were being tagged down here in South Puget Sound in the Nisqually River with acoustic tags, which could be heard on these receivers, and these are the receiver lines in red. And they were able to track them moving through the Puget Sound. And generally, steelhead don't spend much time in Puget Sound. They're in and out in about 7 to 10 days, so they make a really rapid transit. And then they were able to estimate how many of those steelhead were surviving to get out to the Strait of Juan de Fuca. And so this is a figure of the survival. There's a gap in their estimates here because they didn't have enough funding for the study. But what I think is really interesting is that if we look at where our high survival rates for those steelhead smolt are and compare that to when we had high anchovy abundance, we see that in the mid-2000s, 2006, very high. And that drops off to less than 10% survival, almost 0% in 2008. And then 2015, 16, 17, we see that bump up again to almost 40% survival. Now, that's a huge increase when you're talking about survival of juvenile salmonids. And our theory is that essentially there have been enough anchovy in the system that all those predators that we see in South Puget Sound are putting their effort into eating anchovy rather than steelhead smolt. And so salmon in general, they're benefiting from forage fish because they're food for salmon, but they're also benefiting from forage fish because they're not being eaten by the things that are feeding on forage fish. So with that, I think we've got a little bit of time left for questions. Yes?
So any correlation at all between fishing pressure and herring, tertiary herring?
So early on, back in the 70s, the fishing pressure was pretty high, and we think that there probably was a relationship to the cherry point herring decline in the late 70s and 80s. And not just the amount of fish being taken, but the different approaches to harvesting herring. So back then, a lot of the effort was on spawning herring, as opposed to now our effort is distinctly not on spawning herring. And there were some techniques used like spawn on kelp, which is where the eggs, which are adhesive to vegetation, would actually be allowed, the herring would be gathered into net pens, and then kelp would be spread through these net pens, and the herring would spawn on the kelp, and then they'd collect the kelp and let the herring go. And at the time, that was thought to be sort of a sustainable approach to fisheries because they weren't actively killing the herring. But in hindsight, some of the fish pathologists have looked at this and said, that's a really good way to crowd and stress a lot of fish, give them a good way to spread whatever disease they have, and then release them back into the general population so that they can be a vector to the rest of their friends. And so we don't do that fishery anymore, and we actually think that might have had an even bigger impact than the regular take fisheries because of the ability to spread some of those diseases.
Is there any evidence of the cherry point herring
That's a great question. We have had some reports from folks in Canada that they think some of the cherry point herring might be feeding in the Fraser River plume. And we've been trying to get samples to verify this. And there has been some suspicions that they might be using other areas, but we haven't been able to validate that. And it's sort of interesting, if we look at some of the West Coast Vancouver Island stocks, they've seen some similar declines to cherry point herring. So that's why, again, I say we don't think it's an entirely fishery-driven thing when we start to see these regional patterns as opposed to just, one stock blipping in or out. But the declines of cherry point herring are definitely quite worrisome, and we've got some projects underway trying to figure out whether or not there's some way to give them a helping hand, so to speak.
With the nearshore restoration projects, are you seeing positive results, and how quickly that you've seen the forage
Right.
fish come back?
Yeah, there actually have been some good documented results now where I think there's several instances where there had been monitoring before and after a project was installed, and they have seen some increase in surf smelt spawning activity in general. I'm aware of one project that was sort of a restoration of a sort of an industrial site where there had been spawning present for quite some time, but the egg mortality rates were nearly 100%, and they sort of cleaned up that site and put new gravel down, and almost immediately the survival rates went up. So sometimes we'll have spawning in bad areas and we can improve the survival rates. So the trick with that is that oftentimes it's just hard to monitor because you have to have a lot of time and effort monitoring before you've done your restoration to really be able to say for certain whether or not that impact is from the restoration activity or just coincidence.
Could you talk about some of the opportunities and the obstacles to working with colleagues across the border? Because so much is so
Yeah.
shared and so similar in terms of environment, but regulations are different and
Right.
management is different.
Yeah, so definitely different regulations and management approach. And another sort of challenge is that the scales are dramatically different. The scale of the Canadian herring stocks is an order of magnitude larger than the herring stocks that we look at. We sort of joke in the office that, if we ask Canadian researchers to come study our stocks, they'd pretty much say there aren't any because they're so much smaller than what they're used to dealing with in the north. And so just being on the same page sometimes can be a challenge because the figures are so much different, which means the methods you can use are so much different. They can sometimes use aerial surveys to estimate the extent of spawning of herring, where we actually have to be on the ground and looking for eggs, and oftentimes we're only finding a few eggs at a time.
But you can pick up the phone and it's friendly.
Yeah, we definitely have some good contacts over there. I think one of the challenging things is that, with all agencies and we have people come in and they establish themselves as experts, and then they might retire, and then we have to build those relationships again. Yes.
Is there a relevant relationship between algal blooms and outcomes?
Algal blooms and
Outcomes of mortality of the.
I can't speak to that inside Puget Sound. I'm sure if we had the wrong type of algal bloom that we would see a negative impact. I'm trying to think of any that we've observed since I've been working here, and I can't think of any directly. Oh, sorry, we got it.
Just two quick questions that are sort of related to each other. For the first question that I had, in regarding to managing the fisheries and specifically enforcing regulations, is that primarily been state fish and game, or is it also like NOAA with all their conservation and then all that good stuff? And my second question was, you said at the Thulicon were obviously no longer big fish because they're endangered. So the follow-up question was, with all the regulations, are you having any interactions with federal agencies besides NOAA, like for example, U.S. Fish and Wildlife?
Good questions. All right. So the first one, with regards to who manages which fisheries, Inside 3 miles off the coast, typically that's state management. There is some co-management also with the tribes, and depending on the particular species, some of the more migratory species like salmon, then the federal government will be involved in that. But generally, we're mostly involved in the regulation and management of those inside fisheries. Once you get outside, we still have some say in those fisheries, but it moves into more of a federal nexus. Remind me, the second question was?
The second question was the level of interaction that
Right.
the Washington Department of Fish and Game has with other federal
Right.
agencies like Fish and Game.
Yeah, so we definitely interact with them, and we actually, so yesterday there was a Eulicon fishery, that limited fishery. We consult with NOAA before we have those fisheries, and NOAA and the National Marine Fisheries Service are our primary contacts with that regard. U.S. Fish and Wildlife manages some fish species, but typically freshwater fish species. So they've sort of drawn a line where the marine is NOAA and the more terrestrial and then also freshwater species go to U.S. Fish and Wildlife, at least from an ESA perspective.
Usually it's been a lot harder to look for sand lance. Have you developed any new methodologies for sampling for sand lance?
Yes. So, so I'm glad you brought up sand lance. I love talking about sand lance because I think they're really underappreciated. I think they're one of the most critical forage fish in Puget Sound, but because they're difficult to study. So if you're not familiar with sand lance, they're called sand lance because they can burrow into the sand. They spend probably more than half their lifetime in the sand. So that means typical sampling methods don't work. They're also about that thick. So even though they're about that long, they're super skinny, so they'll pass through most nets. They also don't have a swim bladder, which means their acoustic return on the sonar is weak, so we don't get a good acoustic signal from them. And they're gone for several months during the winter, but they're not gone. They're actually burrowed into the sediment. So we're actually using that last one to our advantage. We've been able to identify several beaches in Puget Sound where the sand lance will actually burrow into the sediment during the high tides and remain there even as the tide goes out. And we'll actually go out in the middle of the night, because in the winter our extreme low tides happen in the middle of the night, with headlamps, shovels, and lanterns, and dig sand lance out of the beach after the tide goes out. And that's been one way that we've been able to get our samples. But we're also collecting some habitat data because we don't think people realize how important some of these sandy areas are, because we have evidence that sand lance have been returning to the same beaches for several decades. Papers from the 80s have gone to the same, or went to the same beaches that we're sampling now, and there's still sand lance there. So there's something special about those beaches that they keep coming back to. Yes?
So this might be kind of out there, but, you know, there's sewage treatment plants all up and down Puget Sound, and their pipes go into the sound. Is anybody doing research on the effect of what's coming out of those pipes on forage fish and anything else?
Yeah, so we actually have a group called TBIOS in our Fish and Wildlife unit that is interested in contaminants. They do several different studies. They have done studies on herring. They do studies on resident Chinook. I think English sole is one of their primary indicators. And so, yeah, they do a lot of that sort of monitoring. I think NOAA is also doing some work on contaminants as well, but generally more related to like oil spills and whether or not those can be detected. But yeah, we do have several groups that are focused on that. I think King County is also doing some research on those as well. Yep.
Okay, great. Thank you.
Thanks, everyone.